Yeah, thank you very much, Kate, for the wonderful opportunity. As you've seen before, I was in, or I am in India at the moment, especially because of AMR surveillance at the Christian Medical College in Vellore. And this is really impressive. The numbers they have here in terms of AMR is absolutely incredible. So it's an honor to be, first of all, part of your board and also to give here a short presentation. And I titled this, It's All About the Network, because you are forming this network now. I would like to showcase a bit work we have done in Switzerland, from local outbreaks to then also international ones. And so I will present work of a Swiss pathogen surveillance platform, which is a platform integrating genomic data from various centers. So I think I don't need to tell you what the problem is. There are multi-drug resistant and hypervirulent bacteria and viruses. They are emerging all the time. And it has an impact on humans, but also on animals and the environment. And so to understand the transmission routes between these compartments is really complex. To model that, for example, mathematically, requires a lot of data, and it's not just the sequence data, which is important, also associated data. On the right side, I just give you a glimpse of a Legionella outbreak we had in my hometown. The circles indicate individual bacteria which were sequenced. You can see here the allelic differences in the numbers. A and B point out air-conditioned cooling towers on rooftops of big buildings. And there was an outbreak where we had several cases, and then the question was, what was the source? And we suspected an air-conditioned cooling tower. And what is very interesting, if we just would have sampled one single strain from this cooling tower, we would probably never have thought that this is the reason. What you can see is there's a huge diversity in terms of huge genetic diversity in these strains in these cooling towers, and they also have a transmission network with each other. So sometimes they're really mixed. And then eventually we found out that cooling tower P was most likely responsible, because we had here 3 cases. I will just put on here the laser pointer so you can see these 3 cases could be linked to this particular cooling tower. What is also interesting, the yellow part, so sometimes time also is important. This is the year, so 2010, 2008. So some strains are, around for more than a decade. So what is required for effective surveillance? Besides the technology, I think is nowadays more easy to get, at least in a high-income country. A database for data sharing, that's also possible. But then when it gets really complicated is legal and ethical frameworks. That's something most bioinformaticians don't like. And then also an active community, because I think it's very important to have this kind of activity inside the community. And then I made here with ChatGPT a little rhyme. We need a platform quick and neat to swap outbreaks data, no cheat or repeat. Sensitive stuff we will share with flair, so germs don't party everywhere. And I think that is really, it captures a bit the essence of what is necessary. So bioinformatician streams are made of technology platforms, high-performance computational environments, and databases to share data. But on the other hand, there are also nightmares, and this is legal and ethical frameworks and board meetings, because people want to be effective, and these are things which might slow down, exchange of data. And I think it's very, very important to kind of address them. And for this reason, because we were facing a series of outbreaks a couple of years back, and genome sequencing became available, we thought about forming this platform, the Swiss Pathogen Surveillance Platform. And this is a secure online platform with the idea to share One Health-related data. So not just data on on humans, but also on animals, and merge the data sets inside this platform. It should be usable for research. So there we would share non-sensitive data, but it should also be possible for surveillance, especially with Office of Public Health. That's our public health authority. And they want to share sensitive data because certain pathogens have to be reported by law, even with the residence address. So we needed a platform which can actually address both things, sensitive information and non-sensitive information. And this is what we then built. And so you can see here on the right side, this graph where SPSP is in the center, so a lab can exchange information with the platform, and it's a really secured platform hosted on a very safe environment. Switzerland has invested there quite a lot to build data stream and data hubs basically within the country, which can share sensitive healthcare related data. And then on the platform itself, you have a standardized bioinformatic pipeline, so everybody uses the same pipeline. And then we share actually data very actively with the international repositories, but there obviously we would only share non-sensitive information. So here you would not have a residence address, let's say. And then we could also generate customized reports with the Federal Office of Public Health, which contains sensitive information. So this is the architecture of the platform. You can see here that on top you have the internet, which is not secured, and you log in over an IP whitelist. You have to identify yourself then with a two-factor authentication system, and then you are in the sensitive zone. And in there you can access the data, you can run the pipelines, and we are currently also building up for some of the researchers. They want to have more, let's say, their own tools. So there is also sort of a playground, let's say, where you can run specific tools, bioinformatic tools, which you wish for. But in general, there's a predetermined route, let's say, how the data is analyzed. I will show that to you in a second. And as said, everything is hosted over this very secure node. It's called Biomed IT. And it was initiated over this Swiss Personalized Health Incentive. Okay, the legal aspects and ethical and governance framework really took us a long time. We discussed that for 1.5 years, and that's a long time. And this was the reason why 1 year, after the pandemic, we were then finally able to share genome sequences on a high scale. Before that was legally not possible. And what we've done is we've generated between all major sequencing centers and private labs, which also do sequencing, a consortium agreement that we exchange data with each other, that we work together on public health with genomic data. We have a data transfer and use agreement, so everybody can access the data and use it, but you're not allowed, for example, to go and publish one's other data. I mean, then we also did an ethical approval. We have 5 ethical committees in Switzerland for every region, let's say. And so we also have an approval there. And so whenever we now publish something which contains bacterial, fungal, or viral genome data, we automatically have an ethical approval, which we can use. So that's also extremely convenient, so that you don't have to ask every time, here I have the next outbreak, and you need to wait actually 3 months for an ethical approval, and you want to be fast, and so we have this ready. Another aspect is the governance board. So we have a different governance board. So we have an executive board, we have a scientific board, which is, I would say, the technology part, where bioinformaticians can exchange, they voice their wishes, they discuss platform adaptations and pipeline adaptations. And so the pipeline, which I will show you in a second, is really harmonized between the different centers, and everybody fully agrees with this. And then we have an advisory board, which also contains of different other stakeholders. So we have hospital epidemiologists in there, we have the public health authorities in there, the veterinary authorities are in there, and we meet once a year to have like a big meeting, everybody in person together. So the pipeline is a Nextflow based pipeline. We have a series of tools which are connected. So as soon as you upload the data, you can either go for raw data or already processed data. Then it's assembled, quality controlled. And then you have annotation and a series of individual tools. I will not go here too much into the details. But what is very nice, because it's modular, we can also quality control it very well, and we can also do versioning of the things, and then rerun, for example, uploaded genomes if there's, for example, a new tool coming out. And so what is also very nice at the end here, when you have these reports, we can share this information with the Federal Office of Public Health, and that's very convenient for them. And for this reason, they also give us money now to develop pathogen-specific tools. So that's the next thing we're going to do. We have a list of priority pathogens, and we integrate them now into the platform. So currently we have data of about 650 species on there. You can see here, I mean, it's typical stuff causing outbreaks, Staph aureus, around 2,500 genomes, Enterococcus faecium, Klebsiella pneumonia, and so on. So it's very diverse what is on there, and some species which are very rare, we only have a handful of things, but we encourage basically everybody who sequences and finds something to upload the data there. And so they do that because they trust the platform that their data is not just immediately, you know, picked up by somebody and then analyzed. So here are the different centers contributing in Switzerland. As you know, Switzerland has 4 language regions, and all the regions are here represented in this platform. And what we also do is we try now to connect to different European partners. So we are part of a VRE surveillance network with Germany and Austria, and that's actually very nice. There are thousands of VRE strains now from these 3 countries, and so we also share our data there. And Germany has started a similar incentive, I would say. So it's called My Genome Surf. And the idea there is that different reference labs share data and can be accessed. And there we're also part of. So just a few words before I come to a more international example. So nationally, obviously, SARS surveillance was a driver for us. I really think that kicked off some of the developments we had, and our workflow was from the population where we had positive PCRs, and the RNA could be sent to sequencing labs. And as you've seen in the map before, there were a series of sequencing labs, and then the data was uploaded to SPSP. And here we got from the Federal Office of Public Health, for every sequenced SARS genome, we got some money. And then the condition was to get money that you need to share your data. And then we generated these automated reports, and we also shared data on international databases, as UK did. I think UK, Denmark, and Switzerland were those 3 countries which shared most data. So you can see here what we shared in terms of the population size. This is pretty nice. We had, I think at the moment it's 170,000 genomes, and no, UK has way more, but for us this is a pretty decent surveillance. We have, beside the fact that we have an internal dashboard where you can then access also more sensitive information, we have a public dashboard which gives you an overview on what is ongoing on the different projects. And now I come to an international outbreak, and that was very interesting because we could apply some of our findings. As you may know, there was a large outbreak of Corynebacterium diphtheriae in 2022 with 350-ish cases across 10 different countries. And so initially we started with Germany and Austria to analyze, and then word spread, and all of a sudden the ECDC was part of it. UK joined, France joined, and many other countries. And then we were 10 countries looking into these cases. You can see here the epi curve from May, where it started to get some speed, and then really in the late summer months and fall, you can see here that there were more and more cases coming in from across different countries. There were 3 different sequence types dominating the picture. Sequence type 574, 377, and 384 were the dominant ones. And what is also very interesting is that we had cutaneous cases, but unfortunately, we also had respiratory cases observed. What was very interesting is a subset, which you can see here. I will also show that to you, I think, on the next slide. No. So here is this subset where we also had erythromycin resistance detected. And that's a first-line antibiotic and really worrisome. And this is why I show it to you here, because of the AMR aspect, which is a bit unexpected, and really is obviously worrisome. So you can see here the details. This is the subset of erythromycin strains in the sequence type 377. And what is very nice is we met there also on a very regular basis, I think every 2 weeks during this hot period. And we had a series of phone calls, and ECDC was then taking over the coordination of this. And yeah, really exchanged a lot about that. And that's what I mean with the active community. Okay, I already come to an end. So what was critical for SPSP? What actually made us successful in this platform? So first of all, we needed to realize that there's a true need for this, and that we benefit not only from data sharing per se, but I call this data sharing plus. So we also share the associated metadata. And I think that's very, very important, that you have the context. Then don't lose the enthusiasm and keep dreaming. I think that's very, very important, that when you have such a platform. So technical solutions can be made. That's easy. You can acquire, you know, the sequencing machines. Everybody has this nowadays. But the framework with the public health authorities was sometimes really, you know, taking time actually to get everything done. And the contracts, I think, are very, very important. And what now helps us is this clear governance structure. So whenever we want to work together, we meet once a month, and we talk about projects, and then we approach this together. So forming trust is a very important aspect when you share data. And become a happy, diverse, resilient family, basically. I think that's extremely important. And we include now in this advisory board everybody: microbiologists, hospital epidemiologists, even, you know, many more people, patient representatives, and so on. And I think when we started, it was very important that we had a use case which was almost guaranteed to be successful. That was MRSA. And so everybody was happy, and we generated momentum. And now we expand on aspects which we need most. So the SPSP platform allows data sharing and of molecular and classical epidemiological data. We strongly follow the FAIR principles, and we try to make the data as interoperable as possible with, for example, harmonized vocabulary. And the next steps are now we gradually expand to different pathogens. Pathogen-specific tools are on the list, and we want to align also with other European and non-European partners. And with this, I basically wish you a great experience and this target at AMR. Here are the acknowledgement slides. I mean, lots of people contributed to this, especially Aitana Neves. I want to mention, she's the operational head of SPSP. And if you have also questions, she's certainly also accessible. So thank you very much for the opportunity again to present, and I wish you a very, very good start.